This Salmonella LAMP method has been incorporated within the FDA's Bacteriological Analytical Manual, both as a screening method in animal food and a confirmation method for any presumptive Salmonella isolates.
The method is rapid, reliable, and robust. Compared to PCR, its main advantages include rapid testing, simple instrumentation, a low false negative rate, and flexible workflow.
Rapid screening for pathogens such as Salmonella plays an important role in quickly detecting a potentially contaminated product and can prevent illnesses or outbreaks in humans and animals.
Begin by preparing DNA templates for the LAMP assay. There are two types of samples used to make DNA templates. To prepare templates from animal food samples, aseptically weigh 25 grams of food sample into a sterile filter bag. Add 225 milliliters of sterile buffered peptone water to the bag and incubate it at 35 degrees Celsius for 24 hours. After the incubation, transfer 1 milliliter from the filtered side of the bag to a microcentrifuge tube. Centrifuge the tube at 900 times G for 1 minute, then transfer the supernatant to a new microcentrifuge tube. Centrifuge the sample at 16,000 times G for 2 minutes, and discard the supernatant. Resuspend the pellet in 100 microliters of the sample preparation reagent, and heat it at 100 degrees Celsius for 10 minutes in a dry heat block. Then, cool the sample to room temperature and store it at -20 degrees Celsius.